Hi there. Are you here for biology tutoring? Yeah. Well, my name is Liz. It's nice to meet you. Hi. Yeah. Yeah. So I am part of the university's team to help out any students who seem to be struggling. Yeah. I'm a recent graduate myself. Yeah. So there are different teams here at the university. I obviously am on the science team, but within the science team there are different streams, they call it. So if you need help with chemistry, you're going to go down the chemistry stream and find a tutor that can help you there. If you need help with physics, you're going to go down the physics stream and find help there. Same with maths, they're over there, and here at this table is the biology stream. So I'm assuming you need help with biology since you came up to me. Yeah, so obviously all of us within the biology stream are stronger at different aspects of biology. So what do you need help with today, so we can help find you the right tutor? Okay, well unfortunately we can only deal with like one concept at a time, because we are quite busy. So if you need help with like, microbiology and zoology, you'd probably have to come back another day to get one of them done. Okay, both of them. Well, it looks like Jenna, who does zoology, is a bit busy at the moment. That person just got there for help with Jenna. But I do microbiology if you want to go through that with me. Yeah? Okay, lovely. So what type of microbiology do you need help with? Because, it's a massive study. Yeah, so, you have your infection biology, you have your biotechnology, you have your food science, you have environmental micro. There's some overlap with chemistry, with enzyme engineering, all of that. So it's a massive subject area. Yeah, so I can technically help with any of them, but there of course are areas I'm stronger in. So I'm personally hoping that you're going to choose infection biology. You are? Oh, amazing. Yeah, so that is my favourite area, and where I'm strongest, so I can definitely help you out today. Why don't you take a seat, and we'll go through what you need to go through and, figure out some strategies to help you get ready. Actually, why were you here? Do you have an exam, a lab report? Exam, okay. That's no problem, take a seat. Get comfortable and we'll get started, okay? Okay, you ready? I'm just gonna grab my notebook here, if that's okay. Just so I can make notes as we kind of figure out what you need to learn. And then I will also be using this to kind of draw diagrams if I need, or any of that sort of thing. Okay? So, oops, sorry to click over. So, why don't you start by just kind of telling me about the class you're doing in this exam coming up, okay? Okay, so it's food microbiology, and then can you kind of explain to me how it's been broken up? Because I know each professor does it a little bit differently. Okay, so you're doing the toxins separate from the bacteria and viruses. Okay, that's fine. Who's your professor? I knew it. They're the only person that does it that way, I swear. Everyone else just kind of like separates it based on the disease. They do that too. Okay. it makes life a little easier. I think they changed their curriculum. They heard some people had given out about it. Yeah, no, it used to be like, this is the disease, this is how it works, and then, okay, this is like, and that, this is the bacteria, everything about the bacteria, and didn't really relate the two very well. You kind of had to go back and do them yourself, but I'm glad that they changed it, honestly. It's gonna make it a lot easier for me and for you going through this, okay? Okay. Any other, have you done like any labs? Okay. Could you just describe like kind of what the labs were like? Okay. Okay, and then so they took the meat and no, okay. Can you explain that to me quickly? I'm a little confused then. Okay, so it was like an investigation, kind of like you were presented, right? Okay, so you were presented with this sample and you're trying to figure out what microbe caused the spoilage, right? Okay, that's grand. Okay, I know exactly where you are. Now, the most important question of all of this is, what are you struggling with specifically? Okay, so you're grand on like the toxin side of it, yeah, and then okay, are you alright with the viruses? Sometimes that can be either people find it really, really easy or really difficult. You're grand, okay, that's fine. Personally, I kind of struggled with viruses a bit. I don't find them as interesting as like archaea or bacteria, so that's sometimes why I find it a little bit difficult, you know. So then today, what do you want to go over? So as you know, like I probably forgot to mention at the start, sorry, these are 20 minutes to 30 minute slots to study. So ours started about 5 minutes ago. So we're not going to be able to hit everything today. Of course, you're more than welcome to come back to me and get more help. I'll give you my card at the end. It tells you what days I'm here, so you can stop by. But realistically, we're only going to be able to focus on like one, maybe two, but probably one in good depth microbe today. So what is the one you're struggling with the most? Campylobacter, okay. Well, you are in extra luck because I studied Campylobacter and I know a lot about it, so this is going to be hopefully great for you, and we'll make sure that I still remember everything I studied, which, you know, could have maybe slipped away a little bit. So I will put this down for the moment. But have you heard of this textbook before? Now I know she is very, very big. It's like 1,000 pages, but this textbook is great for just basic knowledge. Have you checked this out one before? No? Okay, that's fine. So what I'm going to do, in it you said you have studied some Campylobacter, right? You have? Okay, great. So what my plan then is to pull up the section on Campylobacter in this textbook, read some of the sentences out to you, and then leave it blank for you to fill in, okay? And then if you get confused on the concept or you need more help, then we'll break it down and we'll do diagrams and all of that stuff, okay? So first, I have to find what page Campylobacter is on. C-A-M-P C-A-M-P-Y-B-A-C-T-E-R Campylobacter. There we go. Alright, so it's on pages 532, and then a lot of it is on page 541 to 42. So I think we're going to go to page 541 and 42. That's okay. So I'll just probably bring this. It probably was just mentioned on those previous pages. So sorry if my hair gets everywhere. Okay. So let's start by what is the order of Campylobacter. So you remember, oh, there goes one of my highlighters. Oh well, be grand. So you remember you have your orders. So there's a little mnemonic that you can use to remember it. So the mnemonic is, I'll say it and then you tell me how they all go. How about that? Great, okay. So it's kings play chess on funny green stools. So each of the letters line up with one of the classifications. So I'll say it one more time, ready? Kings play chess on funny green stools. Okay, so king, which is this? Right, kingdom. Then B. Phyla. What's the next one? Starts with a C. I think you're right. I think it's class. How bad would it be if I got that wrong? I'll have to check that later. So, kings play chess on, which starts with an O. Order, there you go. F is, you got it, family. So you have kings play chess on funny. You need two more. On funny green, genus, right. And what then would be the last one? Species, yes. So this is probably the easiest way to talk about this. Let me just grab my highlighter. So when we start, I'm so sorry, my stuff is banging around. When we start with kingdom. So your kingdom is going to be, you probably have heard it, you know, like your animal kingdom, right? Or your plant kingdom, right? So that's the ground off from your kingdoms. But this can also be, in terms of microbiology, you'll have your archaea, your bacteria, and your viruses, right? So just start with the kingdom. That's what you're looking at there, okay? Okay, let me just double check with Campylobacters. So I make sure I get this right. Again, very embarrassing for me, and it would be very bad for you if I told you the wrong information. Right? Okay, so just give me 2 seconds if you don't mind to make sure that I got this right. Okay, yes. So I think I got it. So then you're going to move into your phyla here. Now, with your phyla, this is where you break up into like all of your different classifications of bacteria. So remember, you start really broad and then you get narrow, narrow, narrow till you get down to just one species at the end. So your phyla, this now, do you remember what Campylobacter belongs to for the phyla? It's one of the most diverse ones. Yes. So they are in the proteobacteria, right? Oh. Proteobacteria. And proteobacteria, can you tell me some characteristics about them? Do you remember? They are all gram-negative bacteria. So let me just swap to a different colour so that can help us with understanding Campylobacter. So we're just going to put gram-negative, encircle that, and draw to the phyla level. Okay, so then we move down into our classes. So remember, excuse me, what can you tell me about the classes of proteobacteria? Yes, there are 5, right? So can you name them for me? So you have alpha, beta, gamma, delta, and epsilon, right? So those are your classes. Now, Campylobacter belongs to one of these. Which one is it? You got this. I'm allowed to look. This one. You got this. What is it? Yes, it is the epsilon proteobacterium. So now that we have that, now we're going to move down into the order. So we also have order and family here, but for time reasons we're going to skip over those. The most important thing is that you remember that Campylobacter, the point of all of this, are epsilon proteobacteria. And then we get down to our genus down here, which is our Campylobacter. Okay? So are you ready to do a bit of a read through now? Yeah, okay. So I'm going to read some sentences out and I want you to kind of fill it in for me, okay? So there are 2 big species in epsilon proteobacteria. Now there are more as well, but there are 2 that this is defined by, right? So one of them is Campylobacter and there's another really closely related one. Do you remember what it is? Helicobacter, right? So just really quick, what is Helicobacter like famous, quote unquote, for? Yeah, so they found it grows in your stomach. It's one of the only bacteria that can survive the stomach in the acidic environment, which is really, really cool. So Campylobacter is related to Helicobacter. Remember that, store that in your brain. Okay, so here's a sentence. So Campylobacter and Helicobacter species are gram-negative, oxidase and catalase positive. There you go. And what is their shape? They both have, they're not the same exact shape, but they are similar. Yeah, they're like a spiral shape. So that's kind of where Helicobacter gets its name from. It's helical, right? Campylobacter, oh, is it bad I don't remember the etymology? Let's just ignore that. When you come back to me, I'll make sure I have it. And how are they motile? Can you explain that to me? So if I were to draw a little really badly drawn Campylobacter here, if I was to do this, how is it motile? Can you explain to me? Is it non-motile? Well, I should start with, is it non-motile? No. Right, I kind of give that one away. It's motile. Right. So, how does it move? No, that's a good guess, but no, it doesn't use the host to move. So it's not like Listeria in that effect. So you know with Listeria, right, it just kind of looks like this, and then it uses the host. So if this is the host out here, it uses the host to push it forward. It's not like Listeria. I know you'd like that one. Think of it as the opposite of Listeria. It has its own mechanism to move. Do you know what that could be? Yeah, it's got some flagella. That's not actually how they look, but it's how I'm going to draw them. Now, it's only at one end. It can be at both ends, right? So, that's how it moves. Oh, something really important about Campylobacter. What is its ideal environment? So is it anaerobic, which means it doesn't like any oxygen? It has to stay away from all oxygen, because oxygen is deadly to it. Or is it aerobic, like we are, where it really loves oxygen and it needs it to survive? Or is it somewhere in the middle, something called microaerophilic, where it needs some oxygen but not as much as we need? Okay, so microaerophilic. So you know, like our atmosphere, right? It's made up of 20 to 22% oxygen. And that can be a lot. That's what we need. And that's what a lot of other animals and bacteria and plants need. But for some organisms, that's too much. So there's something called microaerophilic, where it needs some oxygen, but not as much that's in the environment, in the air. So it needs a very specialized environment to survive. So that's microaerophilic. Right, so it needs some, but not a lot. Okay, so if you need to think about it and figure it out, I want you to think about Helicobacter, right? Campylobacter and Helicobacter are really closely related. And where did I say Helicobacter lived? In the stomach. So that means, right, it's really acid resistant. That's what the cattle is. But it needs to enter us, right? Like we ingest it. It's a foodborne pathogen. So it needs some oxygen to survive. It's not aerobic and it's not anaerobic. But in the stomach, is that like a really rich oxygen area? No. So what would it probably be then? Microaerophilic, yeah. So I think, if I remember correctly, Campylobacter likes about 5 to 10% oxygen, which is less than what's in the air. So it's a microaerophilic organism. So yeah, so it says here 3 to 15% O2, so I was on there, and high carbon dioxide. So that would be higher than we can handle. So that's again textbook. This is 3 to 10%. Okay, we're almost done with this section, and we're almost out of time. So we'll run through the rest of this, and then I will let you be free. And hopefully that this helped. You know, come back if you need it, of course. So, can you describe to me the type of infection you get with Campylobacter? Rather, let's start with how do you get infected with Campylobacter. Now we did the basic physiology bit. Yeah, so how would you get Campylobacterosis? Campylobacterosis, that is the disease caused by Campylobacter species. So it's a foodborne disease. So you'd need to ingest it, normally through contaminated food or water, right? There you go. So it's what's called the fecal-oral route of transmission. Sounds yucky, it kind of is yucky, which means somebody who is infected with Campylobacter expels it from their body, and that somehow gets into water or food. But you know, it sounds gross, like if we're not in the Middle Ages anymore, we have sanitation. That's right. But the interesting thing about Campylobacter, that's how it can spread between humans, is most transmission is from zoonotic origin. Yeah. So it definitely comes from an animal that we eat. But do you know what that animal that we eat most often is that gives us the disease? It's chickens. So the biggest cause of Campylobacterosis worldwide is undercooked poultry. So make sure that when you cook chicken, it's at the recommended temperature and it is fully cooked through, because otherwise you can get sick. And can you kind of describe to me the symptoms? So a lot of foodborne diseases have similar symptoms, so you should so you should have some vomiting and diarrhea and otherwise unhappy tummy, intestines. So is it watery or bloody? Bloody, yeah, it's often bloody, but there's a really wide range with Campylobacter. Now, do you know what the most common cause of Campylobacterosis is? There's one species that causes about 90% of infections in humans. So it's Campylobacter something. Jejuni. So it's Campylobacter jejuni is the most common cause. Okay, so we're nearly out of time, so let's quickly recap what we talked about. So, what is the order to get down to Campylobacter? So it's kings play chess on funny green stools. So, kingdom, phyla, order. Oh, I messed that up. Kingdom, phyla, class, order, family, genus, species. I remembered, but I'm trying to do it in my mind at the same time. What does Campylobacter fall under? What is it called? It's one of those 5 categories we talked about. Yeah, epsilon proteobacteria. Is it gram positive or negative? Good. And how does it move? Yes, good. It's got this flagella. And what bacteria is it really closely related to? Correct. Okay. How do you feel after our little study session there? Better. Okay, I'm really glad. Oh, there you go. There's my card. So as you can see, I'm here 3 days a week, for about 3 hours at a time. So if you ever have need any more help with like Campylobacter or another one or any type of other microbiology question, I'm here. But if you didn't like me, there are other microbiology tutors that will be here that you can come and talk to. Okay. Fantastic. I'm really glad. It makes me happy knowing I could help